Thank you, Derek, for inviting me. It's an honor and a privilege. It's been a great meeting so far. Hopefully that continues. I apologize for my vague title. I had a moment of indecisiveness. I will be talking primarily about OGSE, which is what I was requested to speak about. But before I get to that, let me just give a little background. So diffusion studies for me began when I started my postdoc with John Gore at Yale in 1997. I actually applied to Yale because I noticed on their website, I was still young enough to believe websites, that they had micro imaging, and I thought I would go do that, but the offer was to come and do diffusion work because that's what was funded. So, you know, I looked through their recent diffusion papers and I thought, well, okay, these guys probably know what they're doing. So I was happy to go there. When I arrived, John handed me a copy of his recently funded diffusion grant, which I still have a copy of. I pulled out the other day, and it's just chock full of great experimental study plans, including the oscillating gradient spin echo measurement. So just to put this in a time frame, he was awarded this in 1996, so he probably wrote it early 96, maybe late 95, and he already had this pretty well thought out plan of studies. You know, it was based on the work of Stepiznik and Callahan, as well as the work of Mitra, and included all the diffusion spectrum stuff we heard yesterday from Yanis Stepiznik, as well as some preliminary data. So, you know, a smart postdoc would have just dove right into that, but I got a little sidetracked onto a couple of functional studies I thought I would just tell you about first. So I started off with a study with Jim Pritchard. I don't know if any of you know him, but he's been around the field for a long time, and he had a setup where you could stimulate the optic nerve of a rat, and then my job was to image the visual cortex to look for a change in the ADC with the arriving evoked potentials. And if I had really good data from that study, it would go here. And maybe if I had been a little more persistent, I would have found some interesting results, such as Denis has found. But alas, I gave up on this pretty quickly. But I don't consider it a failure because I got to know Jim, who's a wonderful guy, and I still like to see him at meetings every year. I also got involved in a functionally related study with Jianhui Zhang. I'm not going to go through the details of this, but he and Sharmila Majumdar had written up some papers just before my arrival, or within a few years, talking about the effect of intravascular susceptibility on diffusion measurements, the idea being that if you have a distribution of background gradients, it will reduce your apparent diffusion coefficient, and you should end up with this monotonic relationship between blood oxygenation and ADC. And so we did some experiments to characterize magnetic susceptibility in blood samples, and then we injected iron oxide contrast agent into rats, and we did some really high-resolution diffusion-weighted imaging, and characterized this phenomena quite well. So it was a very fun study from an experimentalist point of view. It is not the kind of phenomena that Denis has been looking at, right, because this is a vascular effect. So the net effect of this paper was really telling you what to avoid if you were trying to do diffusion-weighted functional imaging. But as an experimentalist, it was a lot of fun. All right, so then presumably I moved on to OGSE. Well, not quite yet. I actually made this slide before Greg made his comment the other day about marrying T2 and diffusion. That was, T2 has always been my first love. So I did a bunch of studies involving T2 and diffusion, and I'm actually still working on those. And I was going to talk about that, but you know, I know that if you talk about diffusion or T2 at a diffusion meeting, I might get run out. Actually, I've been surprised at how much interest there's been in T2 here, so I've been pleased to see that. But there's no time for that right now. So anyways, OGSE. So fortunately, having Yanis Stepiznik talk yesterday means that I don't really need to say a lot about the background of the theory, and in fact, it's been discussed many times today already. Gross and Kosfeld had what I think is probably the first paper discussing OGSE. It was a little one-page German-written paper. I used to have a copy of it. I couldn't find it. But the basic idea was just that if you can do a pulse gradient method, and this is your diffusion time, kind of inaccurately labeled there, and you wanted to shorten those diffusion times, you might just run a whole bunch of these together, and that kind of looks like a sine wave, right? So that was their idea, is just do a sine wave, and that gets you short diffusion times. But then Stepiznik wrote up a series of papers that really developed the theory on this comprehensively. And I won't go through this again because he talked about it yesterday, but real briefly, right, it's the idea that if you have free water, your velocity autocorrelation function is a delta function, and so your diffusion spectrum is flat. But when you have restrictions, you get this change in the diffusion spectrum. So that's what we're trying to characterize. The important paper that he also talked about yesterday that is relevant for this talk is that in '95, he and Paul Callahan worked together and proposed the idea that by changing your gradient waveform, your effective gradient waveform appropriately, you can avoid sampling the center of that diffusion spectrum, because that's not where the information is that you probably want. That's like your infinite diffusion time domain. So I'll come back to that idea in a minute. So anyways, the first OGSE study that was published from Yale was done by Melanie Martin. Her name then was Melanie Schachter, and she did sine wave OGSE in these glass bead phantoms with the objective of doing surface to volume ratio calculations. That has also been introduced, so I'll skip over that. Because it was water, we were able to use pretty long echo times. We didn't have particularly strong gradients, so we could only go up to about 87 hertz. So it worked pretty well for the large beads, didn't work so well for the really small beads. But it was a good study. The question for me at that point was, okay, how do we go from here to in-tissue studies? Because we certainly need shorter TEs, we need higher frequencies, and so we're going to need larger gradients. Well, around the time that we were writing that manuscript, I was writing an application, a proposal to the Whitaker Foundation for a grant, and in the summer of 2000, I was awarded that grant, and that money burned a hole in my pocket quickly, and I used some of that to buy these gradient inserts for our Brooker system. So they were 60 millimeter diameter, these little handheld jobbies that had 1,000 millitesla per meter gradients, but they were unshielded, so it took a lot of time doing pre-emphasis work. They were hand mounted, literally, you just slid them in and you cranked this one little knob and these little feet came out and that was supposed to hold it in place. Well, naturally that didn't work really well. So we had to do a lot of packing in and whatnot. So there's a lot of futzing around trying to get this to work, but eventually we got B values up to 400 with sine frequencies up to 1 kilohertz with echo times down to 82 milliseconds, which I considered that was good enough. It's time to get on with the experiments. Ed Parsons, I want to give a shout out to him. He's kind of the unsung hero of this study because it was he who suggested to me that, hey, we should look at this paper from Callahan and Stupiznik in '95 and we should do something like that to try to do this selective sampling of the diffusion spectrum. So thus the apodized cosine waveform was born. Right, so just to remind you, if you do a sine waveform, right, then its time integral has this DC offset, so you're sampling the center of the diffusion spectrum. But by just shifting it to a cosine waveform, you get rid of that. So that's what we did. Ed actually wrote a really nice theory paper that came out a few years later. I don't know how it came out in 2006, because Ed graduated in 2002, but that probably just reflects how slow things are getting done every now and then. But this is a great paper if you're interested. So then finally we ran this study in normal and globally ischemic, right brain globally ischemic, that's dead, right? That's if you don't have a focal ischemia model in your lab and you still want to look at ischemia, you just kill the rat. So Dimitri has already shown this figure, which was very nice of him. I have to say that when we were getting this data, I was just hoping to see some measurable change with frequency, right? We really didn't know what to expect, because I think at that point in time, there wasn't a lot of, I don't think there was any in vivo data that went down to diffusion times that were this short. So we weren't really sure what we'd get, but we were happy with these data, and we had some kind of hand-wavy discussion points. But for the most part, the point of this paper was just to present the data. So it warms my heart, my experimentalist heart, that Dimitri has been able to make quantitative use of this. But going on, from that study, we couldn't, the resolution of the images wasn't good enough to segment white matter, but obviously we had interest in that. As Greg started with his paper that he claims wasn't influential, but in fact began really the modeling of white matter diffusion signals. You had to, if you wanted to be a cool kid, you had to model your signal at some point in time. So we started looking at that. Junzong Zhu, who has been doing a lot of the OGSE work for the last decade, worked out, using the formalism of Stupiznik, analytic descriptions of the cosine waveform signal for parallel plates, spheres, cylinders, the whole 9 yards. And it's a complicated equation, but it's straightforward enough. And this is the result for cylinders, which we validated with computational modeling. So great, that works well. And then we decided to try to test it out with some tissues. There was a bit of a time lag between getting there, and once again, this is just everyone busy doing other things, and eventually we got back to doing some white matter studies. So I had been doing some spinal cord studies for another project, And so I knew it was a great model for looking on axon diameters because the spinal cord is organized functionally, and so you have these tracks like the dorsal cortical spinal tract. This is a rat spinal cord. If it looks different than a human spinal cord, that's why, right? Dorsal cortical spinal tract is very small axons. Vestibular spinal tract over here has very large axons. To give you an idea of the actual dimensions we're talking about here, keep in mind that even on the small end of the rat spinal cord axon size, it's still kind of on the large end of brain, right? So that makes this problem a lot easy. But that's a good place to start, right? So then we developed what I call OGSCaliber. This is my homage to Yaniv, right? Junzong wouldn't let me use that in the paper. But it's a two-pool model, the same as the OGSCaliber, the charmed model, right? Where you have your complicated equation here that I didn't show you. This incorporates all of the characteristic of the axon as well as the pulse sequence. And then your extra-axonal environment. This is a little different than with charmed because we have this frequency-dependent term, or in other words, a diffusion time-dependent term. And this makes sense if you're at short diffusion times, which we are. You're not at that tortuosity limit, so you have to include this. We started off by validating this with histology. So we got histology, and then we segmented it and ran computational simulations based on this histology, and that allowed us to separately evaluate both the intra and extra-axonal compartments, which was kind of a nice intermediate step, and it worked pretty well. And then we did some ex vivo MRI, so you can see we got a whole bunch of different images at different frequencies, different B values, and it fits, the model fits the data quite well across all these tracks. More importantly, when we pull out from the fits the axon diameters, it actually does quite a good job of characterizing axon diameters, even when you consider accuracy, right? So this red line here is the 1-to-1 line. So it's not perfectly accurate, but more accurate than other approaches, perhaps. And we're a little more capable of getting down to these small sizes because we have the short diffusion times. And there's a corresponding map of these axon diameters. And you can really see here this dorsal cortical spinal tract. It may not have been characterized perfectly accurately there, but it certainly shows up as being smaller axons. We also followed this up with what I called the OGSCaliber Lite. Also, I was not allowed to use that in the paper. But the idea is to try to go to a model-free approach, right? Because the previous slide showed data. Yeah, it was an ex vivo sample. We collected a whole bunch of images, and I can't remember how many hours it took, but it was a while. So it's not super practical. So we tried using a slightly more practical approach where we just looked at the rate of change of ADC as a function of frequency. And it's interesting that if you choose the right frequency range to match the axon diameters within your sample, what you see is that that rate of change, so the slope of each of these curves here, changes approximately linearly with axon diameter over a finite range of axon diameters. So if you tune your measurement correctly, you can get a nice correlation between this. So the y-axis here is that slope, and the x-axis is axon diameters, again, from the same sample, and a pretty strong correlation. So it doesn't give you a measure of the axon diameter, it gives you a correlative measure, but it only took 2 acquisitions to do that. So it's an appealing approach. We tried to move this into humans. It's less compelling, but you know, the results, if you looked across 6 subjects, there was a trend that was kind of the correct trend. I don't think we're ready to go there yet. I think the problem with looking in human brain is gradient strength. It's as simple as that, right? So for the apodized cosine waveform and say 100 millisecond echo time, so 2 oscillating gradient waveforms of 50 milliseconds each, be 400. On a mouse scanner, right, you can get up to 500 hertz, which is more than enough. But on a human scanner with 80 millitesla per meter, you're only going to get up to about 40 hertz. And this is a plot here showing those ADCs measured from the rat spinal cord. These are 4 different tracks. I'm sorry they're not labeled here. But the red track here is that one with the really large axons. All the other ones are kind of a little closer to a human brain. And if you look at the slopes down here at 40 hertz, they're all pretty close, right? So you're probably going to have some trouble there. I know what you're thinking. You're thinking, well, we're not limited to 80 millitesla per meter because we have connectome gradients, right? But if you are stuck to the same slew rate, then doubling your gradient means it takes twice as long to get there, which means you aren't increasing your frequency. So there might be some room for improvement with the connectome gradients, but not necessarily a whole lot. That's where connectome, where's Thomas? Connectome 2.0 with higher slew rate gradients would be the thing. So good luck. I don't mean that in a discouraging way. Certainly, when I showed these same data to Christian Beaulieu a few years ago, and he promptly ignored me and then went and got some great data. So, I think that there's probably stuff to be done in human brain. But really, I think that the future of OGSC, I focused this talk on brain because this is a brain place, right? But I think there's a lot more work to be done in cancer. I'm not involved in that myself, but this is some data that Jun Zhang presented at the Lisbon meeting. And this is, I'm not going to go through the details on this. This is just to show that he's had a lot of success in quantitatively characterizing cell sizes in tumors. Okay, in the last couple of minutes, I just want to talk about some of the stuff that I'm doing right now outside of OGSE. So this is my prerogative to just advertise a little bit of things going on. And I have this interest in, I know everybody else in the world is interested in human brain imaging. I am interested in mouse brain imaging, right? I have this idea that there's no reason why neuroscientists aren't using MRI as just a standard tool, that every day when they leave the office, they aren't dropping a mouse brain or 2 or 6 into their little MR scanner and coming back the next day with volumetric parameter maps of measures of interest. So that's kind of my dream. And we have a project right now where we're writing pulse sequences, analysis codes, collecting histology on a bunch of different animal models, and the idea is to create a resource that everyone can use. So, we're using different genetic models of mice that have different characteristics of white matter. We're doing some fairly tedious, and when I say we, I mean students of mine, fairly tedious, exhaustive, quantitative histology to evaluate them. And then we've got results so far that are pretty promising. So these are myelin volume fraction measurements that we make with MR, and these are myelin volume fractions we measure with histology. Not doing quite as well with G ratio measurements, but close. And the G ratio measurements actually involve diffusion, which justifies me talking about it here. And that's it. I just wanted to acknowledge the many people who contributed to these slides. I probably missed a few, but in particular, John Gore has been leading this effort on OGSE with our group for the last 20 years. And for the last decade or so, Jun Zhang Zhu has been doing really most of the heavy lifting. So it's mostly been their work that I've been showing. Thanks.
Thanks, Mark. We've got time for a couple of questions. Then
You're going to ask me about that functional diffusion study, aren't you?
Not at all.
Oh, good.
No, great talk. And I think you should not underestimate the earlier work you've done showing the effect of local susceptibilities to the measurement of diffusion. As you know, we've shown that this trick might give us the opportunity to estimate the content of iron in the brain. So, which is also a parameter for microstructure. So I think maybe you should look back at your earlier studies. They are really interesting too.
Well, so believe me, from an experimentalist point of view, that was a lot of fun doing that study because it was very tedious, but it was nice to get expected results that fit with theory.